Hello and welcome to my YouTube channel, AnatomyMentor. I hope you're all doing well. Today, I'm going to be discussing with you the topic which is the histology of the small intestine. So here I'm going to start with the learning objectives for this topic. So today we will be discussing the general histological characteristics and functions of the layers of the wall of the small intestine, as well as we will look at the important differences between the three parts of the small intestine, namely the duodenum, jejunum, and the ileum. Also, we shall correlate these histological characteristics with the clinical conditions, which are celiac disease and Crohn's disease. Okay, so when we talk about the small intestine, first we're going to look at the dimensions of the digestive tract. You should be well aware of the fact that our digestive tract is in a total of about 9 meters in length. And this is the measurement in the adults. And within this 9 meters, the contribution of the small intestine is the greatest. So the small intestines, they are about 5 meters, or we could simply say the range is 3 to 5 meters long. You know, the function of the small intestine is digestion as well as absorption of the products of digestion. If we want to look at the basic format of the layers, remember the formula of 4, that there are the 4 layers present in the small intestine from the inner to outer aspect within the wall of the small intestine. That is the mucosa, submucosa, the muscularis, and the outermost layer is the serosa, or in some cases it is the adventitia. But there are certain special modifications which increase the surface area for the absorption of nutrients. So what are these modifications within the small intestine? Well, the very first is the plicae circularis, also known as the folds or valves of Kerckring. They increase the surface area for nutrient absorption about 3-fold. Then come the special modifications known as the villi, and they further increase the surface area about 10-fold. Also are present the microvilli, which increase the surface area for absorption to about 20-fold. There is also an additional contribution to an increase in surface area by the intestinal glands, also known as the crypts of Lieberkühn. So when we combine all of these different factors, this gives us a total absorptive area of over 200 meters square in the small intestine. And this area is so large, it is so massive that it can be compared to about 3/4 the size of the tennis court. And that is, it is spread out in such an area. If you would be able to spread it out, that's how large it would actually be. Now let's talk about these modifications one by one, starting with the plicae circularis. Well, the plicae circularis, remember, are a series of permanent circular folds, and they're consisting of the inner 2 layers of the wall of the small intestine, namely the mucosa and the submucosa. Now here, the word to be kept in mind is the word permanent, because this is to be kept in mind when we talked about, if you recall from the lecture on histology of the stomach, we talked about the rugae. They were the folds which were visible on gross examination, or when we look with our naked eye, we saw the rugae in the stomach. We also see such folds on gross examination within the small intestine, which are the plicae circularis. But the difference between the rugae of the stomach and the plicae circularis of the small intestine are that the plicae circularis are permanent circular folds. Whereas if you recall, when we were talking about the rugae of the stomach, over there, those were longitudinal folds which disappeared when there was distension of the stomach. But keep in mind, the plicae circularis, they do not disappear. They are the permanent circular folds which are present within the small intestine. And they also give a velvety appearance to the small intestine from the inside. And they're most prominent in the jejunum. So here you can see that the black arrows are pointing towards these plicae circularis, and they are the transversely oriented folds or ridges, which are filling the lumen up to about 1/2 to 2/3 of its internal diameter, because their extension is partial around the lumen. If the extension were complete, that would have occluded the lumen. So remember, they are permanent transverse folds partially extending within the lumen of the small intestine. Now here is a histomicrograph in front of you taken from the jejunum. This is the hematoxylin eosine stain. You're looking at it through the lower power of the microscope. And here you can see over here, the pointer, if you just follow the pointer over here, this whole area is one plicae circularis. And this constitutes the innermost layer, which is the mucosa, and then the layer beneath it, which is the submucosa. So plicae circularis consists of mucosa plus submucosa. And then external to that comes the muscularis externi. Here is another histomicrograph. This is taken from the ileum, a similar hematoxylin eosine stain section, and we can see the plicae circularis. This is one plica circularis, and then the other one next to it, and then the other one, and so forth. Now, the plicae circularis, I told you, are comprising these two layers, mucosa and submucosa. So this here is the mucosa, which consists of these villi. And beneath it, this here is the submucosa, which has the connective tissue and the blood vessels. And then comes the muscularis externa, which in the small intestine consists of a characteristic inner circular smooth muscle layer and an outer longitudinal smooth muscle layer. And external to that is the serosa, and the serosa is the mesothelium, which is a layer of flattened or squamous cells, and they are supported by delicate connective tissue fibers. Now, the second special modification, which was increasing surface area for the nutrient absorption within the small intestines, they are the villi. And when we talk about the villi, remember their dimensions, they're just about 1 millimeter in length, and they're the outgrowths of the mucosa, and they project into the lumen of the small intestine. Between the villi, there are the openings or in-depth invaginations, which are known as the short tubular glands called intestinal crypts of Lieberkühn. And they additionally increase the surface area for the nutrient absorption. So here we can look at these structures over here in the diagram. Over here, this is a part of the small intestine, and these are the characteristic 4 layers which have been opened and shown to you from the inner to outer aspect. These are mucosa, submucosa, and the muscularis, and then the serosa. And if we were to take a section from here and we were to enlarge or magnify that section, we can see that on the inner aspect, the permanent folds which are visible, these are the plicae circularis. And innermost mucosa is further having these finger-like projections into the intestinal lumen. These are the villi. Now let's highlight or magnify a single villus. And here now in this inset, you can see a single villus, and it is lined by these tall columnar cells, which are also known as the absorptive cells or the enterocytes. Between these cells scattered are the goblet cells, also visible. Well, let's look at this inward or invagination of the epithelial lining or surface. That is the intestinal gland or the crypt of Lieberkühn. So the villus and the crypt of Lieberkühn, they're further increasing the surface area availability for absorption. So the numerous cells, the largest cells in number within the epithelial lining of the villus, which is simple columnar epithelium, are the absorptive cells, which are the enterocytes. And in between them, the second number of cells, which are large in quantity, they are the goblet cells. Now, if we talk about the structure of a villus, we would see internally it has the core of lamina propria. Because if you remember, there were 3 layers of the mucosa from the inner to external aspect. That is the epithelium, the lamina propria, and the muscularis mucosae. So the villus is lined by the epithelium, and within the core of the villus is the lamina propria. Now, this lamina propria also contains some essential structures like fibroblasts. It has smooth muscle cells, it has lymphocytes and plasma cells. It also has blood capillaries and 1 or 2 lymphatic capillaries, which are known as lacteals. Also remember, within the muscularis mucosae, it is supplied by the submucosal or the Meissner's nerve plexus. And the fibers of muscularis mucosae, which is the innermost layer of the mucosa, extend into the core of the villus, and they are responsible for producing the rhythmic movements of the villi, which increase the efficiency of absorption. The muscularis mucosae also produce local movements of the plicae circularis, and this helps in propelling or moving the lymph from the lacteals into the submucosal and mesenteric lymphatics. Now let's discuss these cells a little in detail. Let's talk about the enterocytes or the absorptive cells in the lining epithelium of the villi. These are tall columnar cells with basally positioned nuclei. And their function, is absorptive. They're transporting substances from the intestinal lumen into the circulatory system, that is the blood capillaries and the lymph capillaries. But they also have a secretory function. Keep that in mind. They are secreting enzymes, which are the enzymes needed for terminal digestion and absorption, plus they also secrete water and electrolytes. Now, the other cell population which you see in the lining epithelium of the small intestine are the goblet cells. Goblet cells, remember, are unicellular secretory glands, we could say, which secrete the glycoprotein mucins, and these then become hydrated to form the mucus, which is going to protect and lubricate the lining of the small intestine. The water-soluble mucin is lost during routine H&E section. So when we look at tissue slides which have been stained by hematoxylin and eosine, the mucin is lost, and therefore this part of the cell, the goblet cell, actually appears empty in these slides. Over here is now the histomicrograph, where we are focusing on the villi. And you can see this here is one villus, and next to it you can see the other villus. These are the finger-like projections of the mucosa into the lumen of the small intestine. Let's look at this villus and its lining epithelium. Here are the tall columnar cells, and this arrow is pointing towards these tall columnar cells, which are the absorptive cells or the enterocytes, which we just talked about. And if we look at this arrow now, it is pointing towards these cells which appear to be empty or they have a washed out appearance. These are the goblet cells. Also, I would like you to focus on this part over here, which is an invagination of the lining epithelium present between the villi. This invagination is the crypt of Libercon, also known as the intestinal gland. Within the villus, within the core of the villus, you can see is the lamina propria. Now, this lamina propria, you can see, has smooth muscle fibers. It has lots of lymphocytes and plasma cells, and it also contains the lacteals and the blood capillaries. Here again are the villi, and this is a picture which has been taken through the scanning electron microscope. So over here we can see this is a three-dimensional picture in which these are the numerous villi of the mucosa, and internally this villus, each villus has lamina propria, and externally it is lined by epithelium. Beneath the villi is the submucosa, then comes externally to that is the muscularis externa, and the outermost layer, the serosa. What is the brush border? There's a prominent region at the apical, that is the luminal ends of the enterocytes, and this region is known as the brush border, also known as the striated border. So when we look at the stained tissue slides of the small intestine, this brush border is visible as a faint line in the light microscope on higher magnification. It actually consists of, remember, the densely packed microvilli covered by glycocalyx. So here is the tissue slide from the small intestine, and it has been stained by hematoxylin eosine, and it is a high power view. And here the arrow is pointing towards this faint line, which is visible at you can see the luminal aspect of the enterocytes, and this is known as the brush border or the striated border. And what is it composed of? It is consisting of the microvilli, which are present on the luminal or apical ends of these enterocytes. So the brush border of the microvilli can be better visualized if we use special stains. So in this stained tissue slide section, the hematoxylin PAS stain, periodic acid shift stain has been used. This has made this brush border a little more prominent, and you can see this E stands for the enterocyte. These are the enterocytes or absorptive cells. You can see they are tall columnar cells, and you can see the basally positioned oval-shaped nucleus, and between these absorptive cells are these goblet cells. So because this is a special stain, so that is why the mucinogen granules within the goblet cell have taken a very dark purplish bluish color, and now they are visible. And if we look at this diagram, this histomicrograph beneath us, the microvilli are even better visible, as well as the goblet cell. And here, these are the enterocytes. So remember, these microvilli are the ones which are visible as the brush border in the routine stain sections. Over here, we can look at a very simple diagram of a goblet cell. And in the goblet cell, you can see it is a cell which has the apical portion which is rounded. And that is why it's called the goblet cell, because the apical portion is round and prominent, whereas the stem becomes narrow, also known as the basilar portion. So this is in the shape of a goblet. You know, goblet is a large cup, and that's why it's called the goblet cell. And if we look at the basal portion, we can see the nucleus, and we can also appreciate there is going to be the rough endoplasmic reticulum, as well as the mitochondria. And over here in the apical portion, we see lots of mucinogen granules. And on the apical aspect, a few microvilli are also present, especially in the immature goblet cells, which are located deeper within the intestinal glands. On the left, we have an electron micrograph. This is a picture which has been taken through the transmission electron micrography, which gives us a two-dimensional view of the enhanced magnification within the goblet cell. We can see these are the mucinogen granules, and on the basal aspect of these granules is the Golgi apparatus, and in the stem region, we can see the nucleus and the rough endoplasmic reticulum. Okay, so now we're going to be looking at the goblet cell a little in detail in a three-dimensional digital model, so we can understand these different parts of the goblet cell. Okay, so here is a goblet cell, and if you look at this goblet cell, let's look at the shape, which I told you is like a goblet, because this is the end which is the apical end, that is the end which is towards the lumen, and this is large and round in the form of a cup. And in the basal portion, it narrows down into a stem, and this is the narrow stem of the basal end of the goblet cell. And this is why we use the word goblet, because it is in the shape of a large round glass, a drinking glass, which has a narrow stem or base. So let's first look at this basal region. The basal region, I told you, has the nucleus, and this appears basophilic in the stained sections, in the stained tissue slides, because of the large quantity of rough endoplasmic reticulum. This is responsible for the synthesis of the glycoproteins. And also you can see the mitochondria, which are present over here. And as we move towards this apical portion, we see the large quantities of these mucinogen granules. And also in the basal portion, adjacent to mucinogen granules, we also see the Golgi apparatus, which is the organelle which is responsible for the packaging of the proteins. And at the apical portion, you can also see a few microvilli of this goblet cell. So this is a three-dimensional model, and it describes in detail the essential histological features of a goblet cell. Okay, so now let's look at the ultrastructure of the microvilli within the brush border. As I told you, the brush border consists of the microvilli, plus also it consists of the glycocalyx, which is the layer present on the external aspect of these microvilli. Here are the longitudinal sections of microvilli. Because this is an electron micrograph, so it gives us a higher level of magnification. We can look within these microvilli, which are present on the apical aspect of the enterocytes. And we can see that within their core, these are the structures which are visible. These are the microfilaments, that is mainly the actin filaments within the microvilli. And on the external aspect, this here is the glycocalyx coat. And if a transverse section were to be taken, then here the inset shows that within each microvillus, you can see are the microfilaments. Also to be noted is this terminal web. Terminal web is a region of the actin filaments which are present in the outer aspect of the enterocyte. And the actin filaments, which are present within the microvilli, are anchored to this here terminal web. So how many microvilli do you think are present on the apical surface of each enterocyte? Well, on an average, remember there are about 3,000 microvilli on each enterocyte. And if we want to remember the dimensions of the microvilli, remember each microvillus is about 1 micron tall and 0.1 micrometers in diameter. And like I showed you, it contains the actin filaments. So here is the same transmission electron micrograph. And now on the right side is a schematic diagram, which is showing us the internal structure of the microvillus. And you can see over here that this consists of the actin filaments, which are the microfilaments. And along with that, some anchoring filaments are also present, like formin present at the distal end or the tip or apex of the microvillus, and fibrin, vilin. These are the different anchoring filaments. And we can also see the myosin, which is present in association with the actin. And here is the terminal web in the distal aspect of the cytoplasm of the enterocyte, which is anchoring these actin filaments of the microvilli. Now, when we talk about what are the functions of the glycocalyx? Well, the glycocalyx has a few important functions, and these are, first of all, that the glycocalyx, it is the layer which protects the internal lining of the small intestine from autodigestion by the enzymes. Another function is that the digestive enzymes, remember, they are anchored in the plasma membrane of the enterocytes, and their functional groups extend outwards and they become the part or the constituent of the glycocalyx. Now, the examples of such enzymes would be peptidases and disaccharidases. So here is the enterocyte, and you can see that these are the microvilli of the enterocyte. And here is the digestive enzyme, which is the enterokinase. And it is anchored over here within the apical aspect of the enterocyte. And this enzyme is responsible for converting trypsinogen into its active form trypsin. And this trypsin in turn converts many proenzymes from the inactive to the active state. And this facilitates the digestive process within the lumen of the small intestines. Over here is a similar diagram, which is showing us these microvilli of the enterocyte. And within these microvilli, you can see these enzymes, these disaccharidases are anchored. You can see the lactase, the maltase, dextrinase, sucrase. And what they're doing is that they are breaking down these disaccharides like sucrose, maltoses, and lactose into the monosaccharides, which are galactose, glucose, fructose. And then this facilitates the absorption or transfer of these monosaccharides into the blood capillaries. Let's also look at the absorption of the triglycerides within the small intestine? Well, you know, the very first step in the process of the digestion of the lipids is that they're going to be emulsified, that is broken down from the larger fat globules into the smaller droplets by bile acids and also by lipases. So these smaller subunits, which are glycerol, fatty acids, and monoglycerides, will be taken up by the enterocytes. Within the enterocytes, an interesting process is going to occur. The triglyceride is going to be reassembled and it will be united with a protein, which is the lipoprotein, to form a structure known as the chylomicron. These chylomicrons are then transferred into the lacteals, and from there they enter the lymphatics. Now, when we talk about the process of digestion and absorption, which is taking place within the small intestines, I would like to mention that there are many bacteria which are present within our gastrointestinal tract. In fact, about 100 trillion bacteria are said to reside within the human gastrointestinal tract. And so many species of bacteria, they are present, but we can group the majority of these bacteria, which are the non-pathogenic, or also known as the commensal bacteria. We can simply say the friendly bacteria, which are within our gastrointestinal tract, into 4 main groups: the bacteroids, the Peptostreptococcus, the Bifidobacterium, and the Eubacterium. And less abundant are the E. coli, streptococci, the lactobacilli, and Clostridia. So remember that our GIT has all of these friendly bacteria, and this flora performs many useful functions in the act of symbiosis for humans. They help in the digestion of the long-chain polysaccharides by using those enzymes which the human body lacks, and therefore those molecules which are now achieved as a result of the enzymes of the friendly bacteria can then be absorbed by the gut and utilized. An additional action of these friendly or commensal bacteria is that they compete with and resist invasion of the gut by the other potentially harmful microorganisms. And finally, they also stimulate the development of lymphoid tissue within the gastrointestinal tract. Now, coming back to our basic histology, we talked about the enterocytes and the goblet cells, which were the cells present in the largest quantity or number within the lining epithelium. But remember, there are also certain other cell types within the epithelium of the intestinal glands. And these are stem cells, Paneth cells, enteroendocrine cells, and the microfold cells. Let's talk about the stem cells. We have mentioned them before when we were discussing other areas of the gastrointestinal tract, and we should know that they are the ones which are responsible for replenishing or replacing the other cell types. So this is the cell population which divides by mitosis and gives rise to all the other cell types. And this is responsible for a rapid epithelial cell renewal rate in the small intestine, with each cell lasting for about 3 to 5 days. Their location is characteristically within the basal region of the intestinal gland that is known as the stem cell niche. Now, the other cell type which you should know about when we're talking about the small intestine specifically are the Paneth cells. This is a cell population which is present in the basal part of the intestinal glands. And when we look at the stained tissue slides with the hemotoxin eosine stain, we can see that the Paneth cells present a basophilic basal cytoplasm, but an apical, refractile, eosinophilic secretory granules present that are the source of identification of the Paneth cells. So these are the pinkish, the highly pinkish refractile granules, and these granules contain the defense substances essential in immunity, that is lysozyme, phospholipases, and defensins. This means the Paneth cells have a role in innate immunity. Plus, they also regulate the normal bacterial flora of the small intestine. The next cell population within the epithelium of the small intestine are the enteroendocrine cells. These cells have been mentioned before when I talked about the histology of the stomach. I told you that these are a part of the diffuse neuroendocrine system, and they are secreting different hormones with different functions throughout the gastrointestinal tract. Now, these enteroendocrine cells also have eosinophilic granules, but their granules are present not in the apical portion, but in the subnuclear, that is, beneath the nuclei basal portion. Examples of hormones secreted by enteroendocrine cells are secretin, serotonin, somatostatin, and cholecystokinin, pancreozymin. So here is the stained portion of the intestinal gland, where you can see the basal portion of the intestinal gland at a higher magnification, and we can see the two cell types which I just talked about, the Paneth cells and the enteroendocrine cells. So first, let's look at the Paneth cells. You can see they have these apical, highly refractile, eosinophilic secretory granules. And this is how we identify these Paneth cells. The basal portion is basophilic. And over here also we can see the enteroendocrine cell. And in case of the enteroendocrine cell, you can see that the apical granules are not present, but the granules are basal. And over here, this is the open type of enteroendocrine cell, which means at the apical end, it is in communication with the lumen of the intestinal gland. And this is what triggers the release of hormones from this enteroendocrine cell into the basally located blood capillaries, which carry away the secretory products of the enteroendocrine cell. Now, the other cell type population is the microfold cells. Here, the M cells, we also call them, the M stands for the word microfold. Now, these are large epithelial cells. They have apical microfolds, and in the basal region, they have invaginations or recesses, which contain the lymphocytes and antigen-presenting cells exposed to the underlying lymphoid tissue through an interrupted or porous basement membrane. Remember that these are the cells which are present in epithelium, covering especially the epithelium which is covering the Peyer's patches, and the Peyer's patches are present most numerous in the ileum. Now here is a summary diagram showing us the basic structure and function of the microfold cells. So if you follow the pointer over here, this here is the large microfold cell over here. You can see on its apical portion, there aren't any microvilli, but you have the microfolds. And this is the portion which comes in contact with the antigens within the lumen of the small intestine. And here within its basal portion, you can see are the invaginations or recesses, which contain lymphocytes and antigen-presenting cells. So as the antigen is now taken up by this cell, it triggers a response from these lymphocytes. You can see the basement membrane over here is interrupted or porous. So these lymphocytes, in a reaction or response to the antigens, you can see they're going to be converted into plasma cells, and then they will be releasing or secreting the immunoglobulins. And over here, the secretory immunoglobulin is immunoglobulin A. So this is how a defense is built up. There's an immune response to the antigens, or you could see, say, the harmful substances or the harmful bacteria present within the small intestine. Okay, remember that when we talk about the small intestine, we have these three separate parts. The initial part is the duodenum, then comes the jejunum, and the distalmost part is the ileum. Now, when we have discussed the four basic layers, we know we have the four general layers, which are from the inner to outer aspect, mucosa, submucosa, the muscularis externa, and then the outermost, which is serosa adventitia. Also, we have seen the different types of cells over here. We have seen there's 6 types of cells over here in the lining epithelium, which are enterocytes, goblet cells, the stem cells, the Paneth cells, the enteroendocrine cells, and the M cells. But there are some basic differences between these 3 parts of the small intestine, which would help you to identify a section, a tissue section of the small intestine, and you can identify whether it has been taken from the duodenum, the jejunum, or the ileum. So what are the points which can help us to differentiate between these 3 parts of the small intestine? Keep in mind, the duodenum, jejunum, and ileum, they have the same basic structure, but the difference is that duodenum has a special feature, and that is the mucus glands within its the mucus secreting glands within its submucosa. These glands are known as the Brunner's glands, and this is a histological feature which is found only within the duodenum. Now, why are these glands, these mucus secreting glands, found only in the duodenum? First, we should know that these are the branch tubular mucus glands, and their secretion has a pH of about 8.1 to 9.3. So it can, because it contains neutral and alkaline glycoproteins and bicarbonate ions. So there are two functions of producing an alkaline secretion in this region. We know that the the secretions of the stomach, they are acidic, and when they enter the duodenum, then that can cause an erosive effect on the internal lining of the duodenum. So the primary function of such a neutral and alkaline secretion would be to neutralize and also alkalinize those acidic secretions which are coming from the stomach to protect the duodenum. Also remember, this creates an optimal pH, an alkaline or optimal pH, which is essential for the action of the pancreatic enzymes. Within the duodenum, muscularis externa has the typical layers of inner circular and outer longitudinal smooth muscle. And when we talk about the outermost part of the duodenum, remember it is the only first part of the duodenum which is known as the duodenal cap, which is intraperitoneal. So the outermost layer is serosa. Whereas the rest of the duodenum is retroperitoneal. And in that case, wherever visceral peritoneum is absent, the serosa is going to be replaced by adventitia. I told you before, the serosa is the mesothelium. That means the layer consists of the flattened cells. And adventitia, in contrast, is the layer of the loose fibrous connective tissue. Here we've got the duodenum in front of us. This is a tissue section from the duodenum. We can see the four characteristic or basic layers are present from the inner to outer aspect. This here is the mucosa, the innermost layer, and external to that is submucosa. Then we've got the muscularis externa, inner circular and outer longitudinal layer. What we have to notice over here is that within the submucosa are the mucus secreting glands, that is the Brunner's glands, and they help us in identifying this tissue section as a section which has been taken from the duodenum. Another identification point is the villi. When we look at the villi, we know that they are present in all the parts of the small intestine. But remember, they tend to be the longest and most numerous in the duodenum. And these villi, they become shorter and lesser in number as we go progressively towards the ileum. Another distinctive feature is that the goblet cells, their number increases as we go from the duodenum towards the ileum. So why do you think the number of goblet cells increases distally as we go towards the ileum? Because as you know, that the water absorption is going to increase as we move distally towards the ileum. In the initial portion of small intestine, the nutrients are primarily going to be absorbed. And as we move distally, we can see that the fecal contents are going to be harder and they can impact within the lumen of the small intestine. So the number of goblet cells increases, and because of that, the mucus secretion increases, and this lubricates the contents of the small intestine. It lubricates the internal lining of the small intestine, and this facilitates peristalsis. And peristalsis is the movement or the propelling forward of the fecal contents from the proximal to distal direction. Now, this is a tissue section taken from the jejunum. Over here, you can see the submucosa does not contain the mucus glands. And we have the four typical layers, the basic layers, inner to external aspect, the mucosa, submucosa, muscularis externa, and serosa. Also, you can see that these villi, these are broader over here within the jejunum. They have become shorter and broader as we are moving forwards. Now, when we come to the ileum, the lymphoid tissue becomes more prominent in the form of structures or collections of lymphocytes known as Peyer's patches. And they are typically present beneath the epithelium where the M cells or microphore cells are present. Peyer's patches are present within the lamina propria as well as within the submucosa, and they are actually a part of the MALT, that is the mucosa-associated lymphoid tissue. So here is a tissue section taken from the ileum, and we can see then again the four basic layers of mucosa and the submucosa and externa, muscularis externa and serosa. And what we can also see over here that these are the large collections of lymphocytes. These are the Peyer's patches which are present. We can see them over here in the layer which is submucosa. And this helps in identifying this region as the ileum. I want you to know that there are two types of movements in the muscularis externa. First is segmentation. This is the movement performed by the inner circular layer. And the other one is the peristalsis, which I just told you is the movement which propels the fecal contents in a proximo-distal direction. This is a movement which is performed by the coordinated action of both the inner circular and outer longitudinal layers of the muscularis externa. And the myenteric nerve plexus is located between these two parts of muscularis externa, providing them with the nerve supply. Okay, so now we're going to talk about celiac disease, or also known as celiac sprue. Celiac disease or celiac sprue. This is caused by an immune reaction to gluten. Gluten is a component of wheat, oats, barley, and rye. Now, what happens is that as a result of the immune reaction to gluten, there is erosion of the mucosal villi. And over here, you can see this is a healthy intestine which contains the microvilli. But in this case, because of the immune reaction to gluten, there's an atrophy or erosion of the villi. And as a result of the erosion of the villi, obviously absorption is going to be affected. So malabsorption leads to weight loss, diarrhea. There may be steatorrhea, that is fat within the stool, and anemia, and that is the decreased RBC count and vitamin deficiencies. So this condition is known as the celiac disease, which can affect the small intestine. Another condition, clinical condition, is the Crohn's disease. You might have heard about that before. Crohn's disease is a chronic inflammatory bowel disease, and it occurs most commonly in the ileum or within the colon. So here you can see the picture which has been taken through endoscopy, and you can see the inflamed internal surface of the small intestine in case of the Crohn's disease. So the signs and symptoms due to Crohn's disease will be due to the malabsorption, that is severe diarrhea, and because it's an inflammation can cause low-grade fever, and there can be bloody stools, and of course, eventually there will be weight loss, and also abdominal pain and distension and tenderness in the right lower quadrant. So if we look at the etiology or causative factors of Crohn's disease, it is multifactorial. There can be a genetic factor to it, and there might be some environmental factors to it. Typically, it occurs in the age, which is 15 to 40 years of age. And later on, because of the malabsorption, there can be dehydration, electrolyte imbalance, and anemia. Complications are infection or abscess formation within the abdomen, or fistulae formation, peritonitis, that is the inflammation of peritoneum. These are the different complications of Crohn's disease. Okay, so now that we have discussed the basic histology, here's a clinical scenario related to what we have just discussed, and I hope you would be able to identify the correct option over here. So I'm going to read out this clinical scenario. That is, a patient presents in the gastroenterology department with weakness, pallor, and signs of malabsorption. And the patient has been diagnosed as a case of celiac disease after biopsy of the small intestine. Now, the following histological abnormality is most likely to be expected in this patient. So we know that in celiac disease, there is a destruction of the intestinal villi. So option A is the correct one in this scenario. So here's the other MCQ. The finger or leaf-like projections in the mucosa of the intestinal wall called villi have which of the following characteristic feature? Now, we've just talked about villi. We looked at their basic histology. We know that within the core of the villus, there is a blind-ended lymphatic capillary, also known as a lacteal. So A is the correct option. Let's look at the other options in this MCQ. B cannot be the correct one because the capillaries which are present within the villi of the small intestine are fenestrated capillaries, not sinusoidal capillaries. C is the incorrect one because the core is of loose connective tissue, not dense connective tissue. And D is also incorrect because we know the lining epithelium of the villi is simple columnar epithelium, not cuboidal epithelium. So A is the correct answer in this MCQ. So if this is a flashcard over here showing us the tissue stained section and which region of the GI tract, after listening to today's discussion, I'm sure you can identify this as the small intestine, in which you know number 5 is representing the plicae circularis. And within the mucosa, these finger-like projections are the villi. And we can see beneath it, number 2 is the submucosa. Number 3 is the muscularis. And you can see number 4 is the outermost serosa. Now, this is another tissue section at a higher magnification. And if we look at it closely, we can see it has been taken from the aspect of the lower aspect of the villi. If these are the villi, then this part over here is showing the intestinal crypts or glands between the villi. So number 1 are the villi, and in the lining epithelium, 2 is pointing towards the goblet cells, which appear to be the empty washed out cells. Number 3 is the lamina propria, which is present within the core of the villus. And number 4, in the basal region of these intestinal glands, we can see these cells which have highly eosinophilic granules are the Paneth cells. And also let's look at number 6, the muscularis mucosae, which is the innermost part of the mucosa. Number 5 is the arrow which is captioning the intestinal glands, okay? And number 7 is showing us the submucosa, in which the small blood capillaries, that is the microvasculature, is present, which are the fenestrated capillaries. Okay, so this ends with today's discussion on the histology of the small intestine, and I hope that this was helpful for you and it helped you to understand the difference in the histological features of the duodenum, the jejunum, and the ileum with a better ability to attempt MCQs related to this topic. So if you like today's discussion on histology of small intestine, don't forget to like and subscribe to my YouTube channel, AnatomyMentor. You can do that by just clicking on the picture which is going to appear at the end of this video. So take care, enjoy your studies, and I'll be back soon with another topic. Goodbye, stay blessed.